welcome you all here for the Master's Defense of Inga Conti-Jerpe. I normally don't do this, but I'm going to take a page out of the Koopman Lab handbook and give you a little bit of background about Inga. I don't normally do this, but because Inga and I are birthday buddies, we have the same birthday, which happens to be today, I decided I would, well, and she also shamed me into doing it, so I will say that. She forced me to treat her differently than I've treated every other student, and that's been constant for the past 5.5 years. Assuming I can get to where I need to be, or not. Hold on. This computer should be A. Hold on. Video. I'm just going to go through all the input here. When you see Inga, let me know. There you go. So, right, 1988? And I want to welcome Chris and Jean, Inga's parents, who provided me with all this ammunition. But they did let slip one thing that I didn't know, and that because of all the beautiful hair, Inga's nickname has been Wookiee for many years. And so I took that and this very formally and go through Inga's life. First day of school, she was an outdoor child who loved sports and had a brief stint as a model before she really did find her passion for science and technology. And anyone who's spent time in the lab with Inga will take these final words of wisdom. It's really not a good idea to piss off a Wookiee. So that's what I will know. Now that we've beaten that joke to death, we'll get back to Inga and at 18 months. And we often hear with our marine biology students that they were water babies, that they loved the ocean from the earliest months. And so we have a lot of views of Inga at the beach at 18 months, and then at 5 years, snorkeling in the Florida Keys and collecting on the beach. And I do notice she doesn't have her collecting permit, a habit that we hope we nip in the bud. We got to here. And then at age 10, a severe existential crisis, ballerina marine biology. And we're lucky that the crisis was averted, and thanks to the good folks in Florida who provide manatees and coral reefs and scuba diving, Inga came back from the ballet world into the marine biological world. Loved high school, and this, by the way, happens to be the catalog cover photo for her high school. But I did see this, and I noticed Inga's awfully happy. And you remember that Folgers commercial? Jim never has 2 cups of coffee at home. That was my first thought. Inga never smiles in the lab. I'm not quite sure where that picture came from. There was a brief Alaska internship where you're taking pictures of sheep out of a plane while vomiting. I heard all about that, and I would consider this the great terrestrial mistake, right? We got away from the ocean, the mountains of Alaska, but then found her way to Cornell, where she worked with Drew Harvell. And those of you who know Drew know she's big into coral reefs, and apparently takes care of coral reefs at Cornell in Ithaca. So has the northernmost coral reef in Ithaca, New York. So there you go. Inga took care of that. And then finally, Inga did graduate from Cornell. And for those of you who are worried that Inga didn't leave her mark on Cornell, I got these wonderful pictures from Inga's parents. But then because I've been chair the whole time that Inga's been here, and I really don't know her, I Googled her. And that's a really interesting thing to do to somebody. It turns out there's an entire website that's a tribute to Inga on the Cornell webpage. So there's a lovely tribute to Inga, and you can see Inga in all her glory at the Harvell lab. So they do miss her there, and we were glad to grab her. And from Google, I learned a lot about Inga. She's at UNCW now. Who knew? Been here for a couple of years. Apparently did some work in the Aquarius undersea habitat. And I suspected this, but she spends all of her time on social media. I didn't know she was a tweeter, or whatever you want to call it. But apparently, if you follow Twitter, even during this defense, at Inga Conti-Jerpe will be very active, I'm sure of that. And Inga's been here working 5.5 years. 2.5. A little bit more. 3.5. No, 3 years. On Gorgonians and feeding in Gorgonians, which is what she's going to talk about. Oh, wait. Now I have to find Inga's slide. There. All right.
So I think I can safely say that exceeded all of my expectations. Thank you very much, Chris. I couldn't have asked for anything more. Thank you all so much for being here this afternoon. I'm going to talk to you today about what I've been working on for the past 3 years for my master's thesis, looking at the heterotrophic feeding of 3 temperate Atlantic octocorals we have here off the coast of Wilmington. So as many of you are probably aware, a fundamental concept of ecology is that of the ecological niche. And this can be defined as the combination of conditions and resources that species needs to survive and reproduce. And the classic marine example of this is in the intertidal zone, we have 2 species of barnacles, one of which can exist throughout the entire intertidal zone and has a high tolerance for desiccation, but as it moves lower in the intertidal zone, it's outcompeted by a second species of barnacle. So its realized niche is much smaller than its fundamental niche. And this is what we know as niche partitioning, where competition between species is resulting in this nice distribution that we see in the intertidal zone. We can apply this concept to biological resources like food, and the classic example here is that finches have adaptively radiated to use different food sources, and this has resulted in changes in their beak morphologies that allow them to use these different trophic niches. So again, competition between species leads to the utilization of different food resources. I'm interested in applying this concept to benthic suspension feeders. And these are organisms that feed on cestin or organic particles in the water column. And this has been, niche partitioning has been looked at among broad taxonomic groups of benthic suspension feeders that have very different morphologies. So it's very obvious from looking at an oyster that it's feeding on something different from a coral. But I'm interested in applying this concept to within one of those taxonomic groups, specifically octocorals. Octocorals have extremely diverse colony morphologies. So here in this image alone, you can see that we have a plume colony formation, a fan colony formation, and a rod colony formation. They can also be fleshed and massy or encrusted. Despite their variation in colony morphology, they're all united by having 8 pinnate tentacles, or 8 tentacles surrounding their mouth that have these pinnules or small protrusions coming off of them. So all octocorals use their polyps and their tentacles to capture their heterotrophic food sources. And so they seemingly have these very similar particle capture mechanisms. These species can dominate both tropical and temperate habitats. So here we have a picture of one of our ledge habitats off the coast of Wilmington. And you can see that both sides of this ledge are dominated by 2 different species of octocorals. These organisms increase habitat complexity, so they grow up and away from the substrate, and this can provide refugia for many other species that live in these ecosystems. And finally, because they're benthic suspension feeders, they play a role in benthic pelagic coupling. And this is, through their filter feeding, they capture particles and move the energy and biomass in those particles from the water column into the benthics. Traditionally, octocorals were thought to consume large motile zooplankton, and so many studies initially used Artemia nauplii as a representative food source. However, several studies began to question whether this was the primary food source for all octocoral species. Several species regurgitated Artemia after initially congesting them, and other species failed to consume zooplankton even though when they were readily abundant in the first place. Particulate organic matter has been identified as another food source for octocorals, and this can include detritus and invertebrate and fish eggs. And most recently, phytoplankton has been identified as a viable food source for these organisms. And this is important because this represents a completely different trophic level that these organisms are feeding on. 2 studies have investigated the idea of trophic niche partitioning among octocoral species. Grosowitz and Beniahu in 2012 compared the morphologies of 2 different species within a genus and hypothesized that differences they saw in the length of the pinnules and the orientation of the pinnules were allowing these organisms to feed on different particles in the water column. And therefore exist in the same environments. The only study that has so far observed trophic partitioning among octocorals was published in 2013 by Casito et al. And they found that 4 different heterotrophic octocoral species in the Mediterranean, one was feeding primarily on zooplankton, one was feeding primarily on detritus, and the other 2 species were switching between resources as they became more and less abundant. So the question I'm interested in answering, or I was interested in answering, is do we see this trophic niche partitioning among octocoral species off the coast of Wilmington in the South Atlantic bight? We have 28 species of octocorals off the coast in the South Atlantic bight, none of which have been found to harbor photosynlionons. So this is an ideal place to study trophic niche partitioning since they have to receive all of their heterotrophic food sources, or they have to meet all of their energetic requirements through heterotrophic feeding. These are 6 of the most common octocorals we see off the coast of Wilmington, and I decided to focus my study on 3 Gorgonian species that were the most abundant at the sites where I was conducting my study. Throughout the rest of the talk, Leptogorgia virgulata will be represented by the color orange, Bisianibia will be represented by the color purple, and Mauricia pendula will be represented by the color red. So my hypothesis is that these sympatric species would be partitioning out food resources, and that we would expect to see trophic niche partitioning. And of course, the null hypothesis is that we would see no difference in the niches of these organisms. So in order to answer this question, I decided to use a 2-pronged approach. First, I performed controlled laboratory feeding experiments where the colonies were provided with 2 representative food sources, modal zooplankton and phytoplankton. In these studies, we only included Leptogorgia virgulata and Bisianibia because aspects of Mauricia pendula's morphology made it very difficult to confirm that feeding was occurring in the flume. The second approach was stable isotope analysis, and this is a great complement to controlled feeding experiments because it allows us to look at in situ feeding, so feeding that's occurring in the field. And it accounts for all the available food types that are available to these organisms in natural conditions. So I'm going to start walking you through my flume experiments. The 2 representative food sources that we used were rotifers, which represented zooplankton, and these are much smaller than Artemia noblei, which are about 500 microns. So they're less likely to be regurgitated by the octocorals. The second representative food source was 3 species of phytoplankton. Both of these food sources were obtained from cultures. The rotifer cultures were maintained at the UNCW aquaculture facility in Wrightsville Beach, and the phytoplankton cultures were maintained at the UNCW shellfish hatchery facility at the Center for Marine Science. The total concentration of food in these experiments was set between 15 and 17 microliters per liter, so about 8 microliters per liter per food source. These experiments were performed in a recirculating paddle wheel flume. So here we have the channel of the flume, and the paddle wheel spins and pushes the water around this channel at a constant rate, which was set to 8 centimeters per second. Both salinity and temperature were maintained consistently across all the experiments, and at the beginning of each experiment, the colony was placed in the working channel and allowed to acclimate to flume conditions until polyps were extended and the colony was ready for feeding. Once that happened, the flume was seeded with the 2 food types, and the flume was allowed to mix for 5 minutes to ensure that the food was distributed evenly throughout the volume of the flume. After 5 minutes, an initial water sample was taken, and the colony was allowed to feed for 2 hours, after which a final water sample was taken. And measuring the concentration of the food types in this initial and final water sample is what enabled me to determine how much the colony was feeding of those 2 food types. The concentrations of each food type were determined using an LISST portable particle analyzer, which measures the concentration of particles in different size classes. So in a single water sample, we were able to measure the concentration of both the phytoplankton and the zooplankton. This allowed us to run 5 sub-replicates using both the initial and final water samples. While the colony was feeding during the 2 hours of the experiment, it was also videoed, and this was to confirm that we could observe feeding behavior of the polyps. Each colony that was successfully fed during an experimental trial was sacrificed and coated in a rubberized coating called Plastidip. And then a control trial was run in the exact same way. And this was to account for any passive particle deposition in the flume that may occur during the experiment. So we used the initial and final concentrations of the two food types in both the experimental and control treatments to calculate clearance rates. And the units of clearance rates are the volume of fluid cleared of particles per unit time. So how many milliliters of water were cleared completely of phytoplankton or rotifers given this 2-hour period. This is a useful way of measuring the feeding of organisms because, first of all, it takes into account the control treatment, so it already is accounting for passive particle deposition. And it allows for easy comparisons between different size classes of food. I standardized this value across colonies by dividing by the total number of polyps. So our final units are the volume of fluid cleared per unit time per polyp. So here we have a video of each species to show you what this feeding behavior looks like. So you can see as particles begin to become more abundant in the water column, these extended tentacles are going to fold into the center of the polyps as they capture particles and bring them to the mouth, which is in the center. This is the second species, Inga, which is a little bit easier to see. And so again, we have those tentacles that are fully extended and ready for feeding. And as the particles become pervasive in the water column, you'll see the tentacles begin to fold into the center of that column. So this is how we confirm that feeding behavior was occurring in each of our experimental trials. When we look at the results, what we see is that both Leptogorgia virgulata and Inga were consuming rotifers and not phytoplankton. And so this indicates that they were both able to consume modal zooplankton and none of the phytoplankton species that were provided for them. There was a marginally significant difference between the amount of rotifers that the two species fed on. And so in order to try to understand this, I measured both the tentacle length and the polyp surface area of the two species. 5 images were taken, or 1 image was taken of 5 different colonies, and 10 tentacles and 10 polyp surface areas were measured in each one of those photographs. And what I found is that Inga has significantly longer tentacles and a larger polyp surface area than Leptogorgia virgulata. It makes a lot of sense that a colony that has a larger area to capture particles on would be able to feed more in a given amount of time. And this has been found in the literature. Lasker et al. In 1983 found a similar trend where species with larger polyps were consuming, had faster feeding rates. However, in 1981, Lasker found the opposite trend. And so species that have larger polyps were actually feeding less. And so it seems as though there are many other aspects of polyp morphology that may be affecting the feeding rates of Gorgonian corals. So the flume experiments demonstrated that Leptogorgia virgulata and Inga both consume rotifers, which represented modal zooplankton. Now we're going to walk through the stable isotope analyses that I did, which included this third species, Mauricia pendula, and was trying to look at the trophic relationships of these organisms in the field. So when we measure stable isotopes, we're measuring the relative amount of heavy to light isotopes that naturally occur in different elements. The 2 elements that we're interested in when we were looking at trophic relationships are carbon and nitrogen. So here we have 2 axes with nitrogen, the nitrogen signature on the y and the carbon signature on the x-axis. And one of the most commonly repeated phrases when we're talking with people who work with stable isotopes is higher, heavier, and lower, lighter. So as we move up on the nitrogen axis, we have a higher amount of the heavy nitrogen isotope. Conversely, as you have lower nitrogen signatures, you have a lower amount of that heavy nitrogen isotope. The same thing can be said for carbon, except carbon signatures tend to be negative. So as you move closer to 0, you have a higher carbon signature and a proportionally larger amount of that heavy carbon isotope. So we can superimpose on these axes a very simple model fluid web. And so here we have the primary producers, phytoplankton, our primary consumers, zooplankton, and an octocoral species that's only consuming one food source. And you can see that all three of these trophic levels have very similar carbon signatures because carbon doesn't change as it moves up trophic level. And so we can use carbon to trace a food source as it moves through a food web. Alternatively, nitrogen does change as it moves up a food web. So you can see that as zooplankton consume phytoplankton, there's a jump in the nitrogen signature of the zooplankton. Same thing with the octocoral. Things get a little bit more complicated when we start to have multiple food sources. And so here we have an octocoral species that's feeding on two different food sources. This larger arrow represents that it's feeding predominantly on phytoplankton, and so its carbon signature reflects very strongly the carbon signature of the phytoplankton. However, carbon signatures reflect the relative amounts that these two food sources contribute to the diet of the consumer. And so we see a slight shift to the right as a result of some feeding on zooplankton. The same thing applies to nitrogen. We see a jump in the nitrogen signature that's higher than both the phytoplankton and the zooplankton. This enrichment or increased nitrogen values as we move up trophic levels is predictable. And so throughout systems, throughout many different systems of many different species, the literature has found that enrichment always falls between 2.5 and 3.5 per mil per trophic level. So every time we move up one trophic level, we see an increase in the nitrogen signatures between this range. So what we can do is we can sample a population and plot their stable isotope signatures on the binomial axis. And because of the relationships I just described, this is a great proxy for the trophic niche of this species. So we can draw a convex hull around this population, which gives us an idea of the total variation we see in this of the species' trophic niche. However, you can imagine that as you increase your sample size, your trophic niche is just going to get bigger and bigger. So in 2011, Jackson et al. Came out with the stable isotope Bayesian ellipses in R analysis, or CYBER analysis. And this essentially fits an ellipse to the data with 95% confidence, which is a much better representation of the trophic niche of this species. And so we can measure this for different species and compare where the different niches fall relative to one another, as well as if we see any overlap and the relative sizes of those isotopic niches. So I sampled 3 branch tips from five colonies of each of the Gorgonians present at 9 sites off the coast of Wilmington in Onslow Bay. These samples were collected between July and October of last year, and no site was sampled more than twice. The apical 1 centimeter of each tip was freeze-dried and homogenized using a mortar and pestle, and 2 subsamples were prepared for stable isotope analysis. The first was unacidified to accurately measure the nitrogen signature, and the second was acidified to remove any inorganic carbonates and measure just the signature of the organic carbon. The acidified samples were fumigated with 12 molar HCl, and we avoided using liquid to prevent loss of any compounds from the sample. These samples were analyzed using a Costech 410 environmental analyzer that was coupled to a thermo delta 5 plus ratio isotope mass spectrometer at the Stable Isotope and Geochemistry Laboratory at CMS. With each batch of samples that was run through the mass spectrometer, we included multiple replicates of 2 international standards. And this allows us to express our stable isotope values as DEL values. And a DEL value is essentially the ratio of the heavy to light isotope in the sample relative to the ratio of the heavy to light isotope in the standard. And this is a way of standardizing our isotopic signatures across different studies. This value is expressed as per mil, and running multiple standards also allows us to ensure that the mass spec is functioning properly and we have very good precision. And so here, our precision was better than 0.2 per mil. All of these values were also corrected for the mass of the sample. So when I ran CYBER analysis on the data, what we see is that all 3 species have similar ranges in their carbon signatures, but we have distinct niches that are driven by differences in nitrogen signature. And so Leptogorgia virgulata had the highest isotopic niche, indicating it's feeding at the highest trophic level. Besianivia had a moderate isotopic niche, indicating it's at a Mauricia pendula had the lowest isotopic niche, suggesting that it's feeding at the lowest trophic level. We can also compare the areas of each of these three isotopic niches. And so it becomes clear that Thesia nivea, each of these boxes represents the distribution of 10,000 ellipses that were modeled to fit each of the species of octocoral. And it becomes clear that Thesia nivea has a significantly smaller isotopic niche than either of the other two species. This is exactly what we would expect for a specialist. So these data suggest that Thesia nivea is pulling out a smaller subset of cestin from the water column than either of the other two species. Now, it was very surprising that we saw three distinct isotopic niches of these organisms because we know from these flume experiments that Leptogorgia virgulata and Thesia nivea have at least one food source in common. And so in order to figure out why we're getting these three distinct niches, we had to look at where the samples were taken from. And so you can see that Leptogorgia virgulata is sampled much more frequently inshore. And the isotope literature tells us that there can be terrestrial inputs on the stable isotope signatures of organisms from nearshore systems. Specifically, Baker et al. In 2010 has actually used the nitrogen isotopes of Gorgonian tissues to trace sewage inputs into coastal systems. So I ran a regression against distance from shore against both isotopic signatures of the octocorals. And within species, there was no significant relationship with the carbon signatures, but there was a significant relationship with the nitrogen signatures. And so we see that the closer inshore that the samples were taken from, the more elevated their nitrogen signatures. This is driving the nitrogen signatures of Leptogorgia virgulata up based on where they were sampled from. So I used the linear relationships depicted here for each species to correct the nitrogen signatures of the Gorgonians and reran cybernanalysis. And what we see is that we now have a very small overlap of the niches of Leptogorgia virgulata and Thesia nivea, which agrees with what we observed in the flume experiments. Mauricia pendula, on the other hand, still has this distinct isotopic niche that's lower than either of the other two, suggesting it's feeding at a lower trophic level. If we look at the areas of these corrected niches, we see that Thesia nivea still has a significantly smaller isotopic niche, indicating that it is a specialist. And Leptogorgia virgulata had a marginally significantly smaller isotopic niche area than Mauricia pendula, suggesting that Mauricia is the most generalist of these three species. In order to confirm that what we were seeing was accurate, I also ran cyber analysis just using the samples collected from the two sites where all three species coexisted. And we see very similar trends. So here, nivea and virgulata have very similar nitrogen signatures, while Mauricia pendula still has that distinct lower isotopic niche. We don't see any overlap in virgulata and nivea here because we only have an N of 6 for virgulata. And when you're running cyber analysis, you need a much higher N, closer to 20, to get an accurate depiction of the size of the niche. So we see this niche partitioning particularly with Mauricia pendula. And we know that virgulata and nivea are at least consuming modal zooplankton as part of their diet. But what is Mauricia consuming? I wanted to try to get an idea of what may be contributing to the diets of these organisms in the field by comparing the isotopic signatures of the Gorgonian tissues to the isotopic signatures of potential food sources. So at each site, I also collected, in addition to the Gorgonian samples, particulate organic matter samples and zooplankton samples. POM was collected by sampling bulk water at depth using a collapsible Canton jug. So the jug was compressed at the surface, brought down to the substrate, and the water was sampled between 1 and 3 meters above the habitat. Particles larger than 20 microns were removed from this sample using a mesh sieve, and the remaining particles were vacuum filtered onto a pre-combusted glass fiber filter. Zooplankton were collected by hand towing a net with a 100 micron mesh, and particles larger than 1 millimeter and smaller than 64 microns were again removed using mesh sieves. These samples were treated very similarly to the Gorgonian samples in that they were freeze-dried, and the zooplankton samples were homogenized. 2 subsamples of each were run again, acidified and unacidified, to accurately measure the carbon and nitrogen signatures respectively. So here we have the same plot that we've been looking at for the stable isotope portion of this talk, and you can see we have the 3 octocoral species plotted in their 3 separate colors. And on top of that, we put the zooplankton signatures and the POM signatures. And we see that the carbon signatures of these 2 food sources are falling right around the middle of where the Gorgonian samples are clustered. And this is what we'd expect since we don't see any enrichment in carbon as it moves up trophic level. These are also relatively low compared to the bulk of the Gorgonian samples, which is also what we would expect as nitrogen is enriched as it moves up trophic level. I ran regressions to make sure to see if we saw the same relationship in the nitrogen signature of these samples along distance from shore. And the only significant relationship that occurred was in the carbon signatures of the POM and distance from shore. However, while this was significant, the slope is very small, and the R-squared indicates that it only explains about 40% of the variation. And so this didn't seem to be an important trend. Now, as you recall, I earlier told you that the enrichment factors for nitrogen are predictable. And so across the literature, every time nitrogen moves up a single trophic level, it's enriched between 2.5 and 3.5 per mil. And so we can use this as a minimum and maximum enrichment if we want to compare the signatures of our food directly to those of the Gorgonian corals. So here we have the same plot, and we have the unenriched raw data for both the zooplankton and the phytoplankton. Then we have the minimally enriched signatures. So here, carbon has not been enriched at all. So you can see that the zooplankton is directly above the raw point, and POM is directly above the raw POM point. And nitrogen has been enriched 2.5 per mil. We also have the maximally enriched zooplankton and carbon and POM signatures. And so here, carbon has been enriched 1 per mil, and nitrogen has been enriched 3.5 per mil. And these points represent the total possible ranges of enrichment for these food sources when they're consumed by the Gorgonians. And so this gives an idea of how we would compare these values to the Gorgonian values. So we can see that Virgulata seems to much more closely match zooplankton. It has some overlap with the high enriched POM. Nivea matches the low enriched POM, and Mauricia is below both of these enriched scenarios. Which is surprising because if Mauricia is consuming either of these food sources, we would expect that it would fall in line with one of these enrichment scenarios. However, there were several issues with this study that make it difficult to draw any conclusions about what these organisms are eating using the stable isotope analysis. So first, I only sampled for 4 months out of the year. So we're missing any annual variation that may be occurring in the signatures of the zooplankton and the POM. Additionally, samples were taken anywhere between 7 and 30 days apart. And because zooplankton and POM have fast turnover, it's possible that there's smaller scale variation that could be occurring in these food sources. This is not an issue for the Gorgonian samples because we use this top 1 centimeter apical length of the branch tip, which represents between 6 and 12 months of growth. So when we sampled that 1 centimeter of the Gorgonian, we were sampling a much longer time range of growth for those species. The second issue that we have with these data is that when we separated out the POM and the zooplankton, we're only separating by size, not by actual food type. So in the POM samples, it's very likely that we have microzooplankton, detritus, and phytoplankton all lumped into one food source. So that can be that is strongly altering the signature of this food source. Finally, we don't know the enrichment factor for octocorals, and that's why we have this range of values. And this becomes important particularly for Virgulata, since it seems to have some overlap with POM, but only in the high enrichment scenario. And so it's very difficult using these data for us to draw any conclusions about what these organisms may be feeding on, based on the isotopic signatures of the 2 food sources. So to summarize what I found, the controlled feeding experiments demonstrated that Virgulata and Nivea both consumed motile zooplankton or rotifers. And a stable isotope analysis supported this in that Virgulata and Nivea had overlapping niches, but we still saw a distinct trophic niche with Mauricia Mauricia pendula that was lower than the niches of the other two species, suggesting that it's feeding at a lower trophic level. This is all despite the fact that all three of these species have very similar polyp morphology, so they still all have those 8 pinnate tentacles that they're using to intercept particles in the water column. So this begs the question, how are these species partitioning out different trophic resources? So in the literature, it's been speculated that pinnule length and the space between pinnules could have an effect on how these organisms strain out particles from the water. Similarly, nematocyst density may affect the size of particles that they're able to capture, and colony morphology has even been speculated to affect the turbulence that these organisms generate, which can create eddies that allow polyps farther downstream in the colony to capture particles. Mauricia pendula was excluded from the flume studies because it was very difficult to observe feeding behavior. And this was because of a high density of surface sclerites that you can see here, this very rugose surface of the colony. And so it's possible that this may be playing a role as well in the feeding of this species. Behavior is another important factor that may be driving selectivity. So different Gorgonian corals have different stimuli that cause them to extend their tentacles. Some extend them during the day, some extend them at night, and so there's different aspects of their feeding behavior that may allow them to target different types of particles. So we see this trophic niche partitioning among species. What does this mean for the systems that these organisms inhabit? And because they play a role in benthic pelagic coupling, we can now no longer treat octocorals as a single functional group. Instead of lumping them together as something that pulls zooplankton from the water column into the benthos, we now need to consider variation that we may see in the feeding of these organisms and how this may affect the systems that they can dominate. On top of this, several studies have found that octocoral populations in many parts of the world are increasing. So here you can see that in the past 15 years, in a Keys-wide survey, the percent cover of octocorals has been increasing to the point where they are have the highest percent cover of any of these major taxonomic groups. And this indicates that as these organisms, while we don't know if this is occurring on North Carolina hard-bottom habitats, we now can no longer just assume that the effect of this is universal no matter which species are making up this increase. So we can no longer treat these organisms as a single functional group in the systems that they inhabit. With that, I'd like to thank everyone who helped me complete my thesis. A lot of people have put in a lot of time and energy to help me understand everything. I'd like to thank my lab group, everyone who helped me collect samples, particularly Dr. Wilson Freshwater, without whom I wouldn't have been able to do either parts of my study. I'd like to thank the Stabilized Stope Geochemistry Lab, who was an excellent resource and really helped me learn how to run my stabilized samples and interpret the data. Dave Hines was a huge help with the cyber analysis that I had to do in R, and the aquaculture facility and shellfish hatchery both never seem to run out of rotifers and phytoplankton, no matter how many times I need to rerun and rerun and rerun my flume experiments. I'd also like to thank my 2 advisors. I've been lucky enough to be co-advised by both Chris Finelli and Joe Pollock, and I really think that I wouldn't have had anywhere near the same experience if I didn't have both of these wonderful mentors to help me through this process. With that, I will take any questions.
So that was a really neat niche experience. Now I know why you asked the question you did at the end of my talk. So going all the way back to your first slide, looking at the difference between fundamental and realized niches, I don't know that you were able to look at it in your study, but if you think about where all 3 may overlap versus what their diets or their niches look like where you just sampled 1, do you see a difference in niche breadth or width when you find them alone or when you find all 3 together? Again, going back to that realized versus fundamental niche idea.
No, that's a really interesting question, and we don't really have, so one of the biggest issues when doing this work is that Virgulata is much closer inshore, and the other 2 species are much more abundant offshore. And there are instances where one site will be dominated by Nivea, and it'll be no or very little or no Mauricia, and other sites where Mauricia will dominate, and you don't see a lot of Nivea. I haven't looked at how the signatures vary between those sites, but I think that would be really interesting to look at. Also, the one other paper that's looked at this, Cosito et al. 2013, did see those two species that switch between food types based on availability. So it's very possible that competition at a single site may be altering what they're targeting in the water column. Yeah, that would be really interesting to look at. Maybe I'll try to include that if I can see it.
For your food study, you're looking at the phytoplankton and zooplankton. You see a positive, but they're eating more zooplankton. Why was the phytoplankton negative?
That's a real, okay, so when we get a negative clearance rate, that means that there's a greater decrease in the control treatment than the experimental treatment. So basically, for some reason or another, we saw a little bit more passive deposition in the control treatments than the experimental. Those negative numbers are really small, and so it's kind of just some of the random variation that's occurring in these experiments. But that's a great question. It confuses a lot of people. Yeah, Gina.
I'm not as savvy about corals, but is there a chemical cue on the polyps when the food passes by that they can say, oh, that's food, and that, I don't like that food. So.
I don't think anybody's investigated that. So they can capture particles using the nematocysts, which are like harpoon-like cells that are triggered through tactile stimuli. So there's no chemical cues involved in the nematocysts. Whether there are other chemical cues, I don't think there's any evidence to suggest that in the literature as of yet. And I think it's an interesting one. I thought about doing that at the beginning of my thesis. But yeah, it's a hard question to answer. Clark.
So I know confirming that Mauricia pedula was feeding was really difficult because it has such a small polyp size. But in my head, it makes sense that it has a small polyp size because it seems like in your graphs, it seems to be feeding on possibly the phytoplankton or very, very small POM organisms.
Mm-hmm.
Did you run flume experiments with Mauricia, and did you see that they were eating phytoplankton, or yeah, that's my general question.
So Mauricia doesn't have smaller polyps. I don't have any quantified measurements, but they are probably smaller than Nivea based on what I've seen, but I don't think they're smaller than Virgulata. What made it very difficult to observe them in the flume is that they have surface sclerites on the surface of the colony. And so the polyps are kind of like tucked partially behind these surface sclerites, so you can't see if they're pulling their tentacles into their mouth. I did run some trials with Mauricia in the flume, and I couldn't tell if they were eating, and I wasn't getting any decrease in particles at all. So whether or not that's due to them not feeding in the flume, or it's due to them not selecting any of the particles I gave them, or it's due to them not being ready to feed or not acclimated to flume conditions, is impossible to tell because of that. That was kind of the trouble I had and why I ended up excluding them from the flume experiments. Yeah.
Sort of like Gina, I have zero idea what this coral, but in your flume study, is there, obviously it's a laboratory testing and it's going to be different from the actual corals in situ. Would depth have any effect on if you have a Gorgonian at 20 meters underneath the surface, you would have your Gorgonian in a little shallow trough, would that have any effect on how on your rates or what they took up, or would light have any effect?
Maybe. That's really hard to say. There's been a study that has looked at the effect of depth on feeding, but that was with a species that had photosynthetic symbionts. So they saw a difference in shallow and deep individuals, but again, it's because they're able to get different varying amounts of carbon from their photosynthetic symbiotic algae. These species, I think you would have to do a lot more convoluted logic to come up with a reason why light and depth would affect their feeding, but I'm not going to say it's impossible. I mean, I think it's very hard to know. And they were feeding in the flume, so that suggests that they can be feeding normally. You would think that if there was something about coming up to the surface and being in a shallow little flume, that they wouldn't eat at all. helps create the turbulence that allow the downstream columns to capture the food, and then the whole colony can survive. Varying levels of turbulence may affect what they're able to feed on, but I think more likely it's affecting how much they're able to feed. That answers your question.
There's no more questions. Well, thank you, Inga, for